Welcome to Neuronal Synapses by me. In this video, we'll be talking about neuronal synapses, chemical synapses in a particular, and we'll also be talking about action potential, which I briefly mentioned in the last video, but I didn't explain what it is. So here is action potential. First, we're going to be talking about action potential, and then about neuronal synapses. For action potential, we have to draw a graph. 5. And for the horizontal axis, it's going to be time, and for the vertical axis, it's going to be potential. First, in red, it's its resting state. It's the neuron's resting state. The neuron's resting state is always, it's not always, but it's when it's not activated yet. It's usually at negative 95 millivolts. But then, when a signal enters through perhaps from another neuron, it will create a signal which creates a graded potential. Not as powerful as an action potential, but small and brief. It electrotonically affects this part of the neuron too. But that part is crucial because then, when it electrotonically affects that part, the axon hillock in specifically the axon hillock, it will suddenly rise up to possibly meet the threshold. The threshold is this dotted line. The threshold is super important because if it passes the threshold, it will enable the action potential to spread throughout the neuron. Third part, the action potential. Once it reaches the threshold, or if and when it reaches the threshold, then it will start activating voltage-gated channels which make the neuron even more positive. This is called an action potential, and it's much, much more effective than a graded potential. It will open sodium channels which make it even more positive, the neuron more positive, and then it will activate even more sodium channels further down the neuron. This is an action potential, but by the time it reaches the other neuron, it would be the sodium channels on one side will already stop. So it goes down all the way back to its resting potential. The peak is at about 70 millivolts. And it's positive, not negative. So the difference is 165. It increases by 165 millivolts. And that's a large number of millivolts. Definitely. So then, the action potential goes all the way through. The myelin sheath makes the action potential travel through the neuron even faster. Let's say to pick up a pencil, you need the action potential throughout one neuron, and then the other neuron, and the other neuron, and one neuron that goes to the muscle, which tells the muscle to pick up the pencil. So, and the myelin sheath makes it go faster. The inside of the cell is more negative than the outside of the cell. The outside of the cell is positive, and the inside is negative. But for every neuron, there is an end. Because the distance from our head to our feet is not infinite. You know that. So for every neuron, there should be an end. Either if it's meters or if it's just a few micrometers. it will always end somewhere. Specifically, right over there at the axon terminal. See the axon terminal? What happens right here when it connects to another neuron? How does it pass? This is why we have the synapse. Now we're going to go to the neuronal synapses part of this video. Now we just need to understand a few technical terms. First we have the presynaptic neuron, the neuron that has the action potential and wants to pass it on the message on to the other neuron. Then we have the postsynaptic neuron, the neuron that needs to receive the message in order to pass it on to many more neurons, or pass it on to the muscle to pick up your pencil that you need to do work or homework or whatever. And then in the middle is called the synaptic cleft, which is 20 nanometers. Very small, 2 times 10 to the 8 nanometers. It's even smaller than a virus. So when the action potential goes through this neuron, obviously it will open up the sodium voltage-gated channel, which fills that part with positivity. Yes. And it triggers more sodium voltage-gated channels. But because this is the end of the neuron, and the end of the neuron has one more type of channel, calcium voltage-gated channels. Calcium has 2 pluses, calcium plus, which will go in and fill up the cell with calcium. But why calcium? Why isn't there just more sodium? Of course, it triggers more calcium gates. For the first part, why calcium's needed, we need to understand that there are vesicles in this double membrane, and this is where we come to the double membrane part. Everything in the human body, every cell has double membrane, which means that the neuron is actually like this. The edge of the neuron is actually like that. But we're just going to draw a single line. For the vesicles, we're going to draw 2 lines. But what do the vesicles contain inside? Obviously, neurotransmitters. These will transfer the action potential to the other neuron, the dendrite of the other neuron. And the things connecting. And how do they get across this barrier? That's why calcium's needed, to help these vesicles dump the neurotransmitter across the neuron. On every vesicle, there is a snare protein complex. A good acronym and a good word, because it's an acronym, also a good word because it sort of snares on for the vesicles. What happens when calcium binds? Let's see. Calcium's supposed to bind here, which changes the shape of the snare proteins just enough to change its position. And now it pulls in the vesicle towards the neuron end. So what happens when it touches the neuron end? This is what happens when it touches the neuron end. The blue things are the snare proteins. And they now create an opening because it binded. And what do the neurotransmitters do? They do their job. Go outside the neuron, the axon terminal, into the synaptic cleft. So now, the neurotransmitters go into the synaptic cleft. And the neurotransmitters activate a sodium channel, but it's not voltage gated, it's neurotransmitter gated. When the neurotransmitter binds to the channel, it will open, allowing sodium to come in. Of course, it might be an inhibitory neuron too to stop the message. If it stops the message, it's a potassium channel, which means the potassium will go out, which is inhibitory, so that it will be harder for the action potential to cross. But we need, in this case, to pick up the pencil, we need a non-inhibitory one, a inhibitory neuron, which starts the cycle all over again of action potential. And then the action potential, the positivity, will go throughout the neuron. And that's why there are neurotransmitters. And that's the purpose of calcium. If there is excess calcium, there's one more thing. What about excess calcium? There's excess calcium, there's this ATPase to protect you from that. There is always one ATP in here, and the excess calcium will go to this ATPase. The calcium entering changes the protein. No, not that. The calcium entering makes the ATP lose its phosphate, which generates energy. The energy basically changes the protein just enough so that its job now is to push out the calcium in it. So then this outside will get full of calcium too. And that's the end of transfer. Now the action potential will go to do on its job, making the person's hand pick up the pencil.